Thank you very much. We move breathlessly onwards. It's really a huge honor to be here, to be chosen as an awardee for this prize with Huda. Humbling, really. My motivation and the way I got into this is very different. Huda is a clinician who saw cases, was intrigued, and burrowed down through the genetics to the molecular side and the circuitry, etc. I'm not a clinician. I started my work as a basic scientist who was just interested in blue skies and certain aspects of DNA function. So this is, if you like, I came from the bottom up as opposed to the top down. I thought I would just mention that the UK, at least a few months ago, was part of the European Union, and we are not closed for business, despite the impression that our strange decision may have created. So I'm going to start in rather general terms and then get more specific and narrow. The body is made up of large numbers of different types of cells, and here are some of them. The different types of cells we now know, we didn't when I started, but we now know have the same genome. And their variable characteristics are due to the expression of different genes. So cells look different because the activity in the different cell types is different. Gene expression programs depend upon sequence-specific transcription factors. They come in, they bind to promoters in combinations. Their abundance and which factors there are determines which sort of genes are expressed, and that determines what sort of cell it is. And so it sort of follows from this that differentiation should be completely reversible. Every cell has all of the information needed to make all of the cell types, so therefore it should be straightforward to make any sort of organism, or indeed the whole organism, from any one cell type. But as you will know, for many years this proved difficult, and people got used to the idea that you could not convert one cell type into another cell type. They were stuck once they differentiated. But experiments from these guys, John Gurdon, showed that he could make frogs from skin cells. It was difficult, and sometimes he had to launder those nuclei through another oocyte in order to get them to do it. Then Ian Wilmot, famously in Edinburgh, showed that you could make a sheep from a mammary epithelium. Again, going backwards, turning back the clock, if you like. And Shinya Yamanaka, of course, showed that you could, using certain procedures, convert most differentiated cell types into stem cells. So there are ways of turning the clock back, but they are still frustratingly inefficient. Once a cell has differentiated, even though it has all of the instructions to make all of the cell types, it behaves as though it is somewhat reluctant to reactivate these genes and start again. And this is sort of illustrated in a picture, the very famous picture from Conrad Waddington. So he was the Buchanan Chair of Genetics, which I now am, so like 4 or 5 editions ago. And he drew this picture in which this is a segment of the egg, and the ball rolls down, and decisions are taken depending on which valley of the epigenetic landscape that the piece of the egg rolls into. And you can see once you're down here, you don't have so many options. You can get over the top into another valley, but it's tricky to do. So the question is, why is it so irreversible? If you take a nucleus from a differentiated cell and you put it into an egg, why doesn't it just go back to taking the instructions from the transcription factors that are in the egg? And the answer is really epigenetic processes that kind of consolidate decisions taken by transcription factors. They also almost glue them into place so they're difficult to change. And these are based on epigenetic marking, or at least partly, and perhaps to a great extent due to that. So what are these epigenetic marks that, as well as transcription factors, are involved in consolidating gene expression programs? Well, here's a set of chromosomes, and if you unravel those chromosomes with, say, high salt treatment or something, you can see that it's beads on a string, and these are the nucleosomes, the chromatin that you know about. And the question is, how do you tell one bit of chromatin from another? And the answer is that there are adaptations in the form of chemical marks that are added or subtracted from these nucleosomes. So in here, for example, you have a piece of the genome, and it's covered in marks that are associated with stable gene activity. Here, on the other hand, you have a region that is associated with transcriptional silence. It's not making, the genes are inactive. So what are these marks? Well, ideally, if you wanted to mark your genome, you'd write on the DNA. But unfortunately, you can't write much on the DNA without destroying its function. The only thing you can do is put on methyl groups on cytosine. Not quite the only thing you can do, but the most prominent thing you can do among multicellular animals is put on this methyl group. And this is more or less neutral. It doesn't interfere with too much. It makes the cytosine actually quite a lot more mutable, 10 times more mutable, and many genetic diseases, including Rett syndrome, are caused very often by mutations that occur at methylated cytosine bases. So, but it's relatively neutral. That's one way of marking, but you need more than that, and it appears that a major function of these chromatin lumps, these nucleosomes, is to have tails on them that can contain information. And that information is in the form of methyl groups, acetyl groups, ubiquitin, phosphorylation, etc., which is added on and constitutes these marks. So epigenetic marks then take the decisions of transcription factors and kind of fix them in place. And that's a plausible reason for why it's so difficult to go backwards and reprogram. So you can define epigenetics as a structural adaptation of chromosome regions so as to register, signal, or perpetuate altered activity states. Epigenetics has other definitions, and for every lab almost, there's a slightly different one, but this is the one I'm going to refer to, and these marks create that epigenetic landscape that I showed you in the previous picture. And these, you may think from what I've said that they are secondary, and secondary things are not important. But actually, they are pretty important for disease, because it turns out that in the explosion of data we now have from exome sequencing, of developmental delay, of cancer, of various other disorders, genes that are involved in writing these marks, reading these marks, or erasing these marks are disproportionately frequently found. And that applies to neurological disorders as well. And so as understanding how these consolidators work is of great importance. So I'm just going to give you my sort of view, and I'm not going to justify all of it, of the way these things work. I'm sort of repeating myself, but gene expression patterns are established by DNA sequence-specific transcription factors. It's not as though, in my opinion, epigenetic marks tell the factors where to go initially. The factors tell the epigenetic marks where to go more likely when the evidence is known. Developmental changes are usually determined by internal logic, largely insulated from the environment. There is a view that epigenetic marks are susceptible to the world we live in, your experiences, how your parents treated you, and what you had for lunch. But in my opinion, the evidence for that is not always particularly strong, and in fact, organisms take quite a lot of trouble to make sure that the environment doesn't affect them, except in cases of heat shock or disease where they need to respond. So the epigenome is set up in response to genome activity as a secondary consequence, and these mechanisms buffer, optimize, stabilize the decisions taken by the transcription factors. So it's all part of one system, and this, despite being secondary, is pretty important. So the area that I've worked on is epigenetic marks, which are added to the cytosine base. Here is a methyl group. It sits in the major groove, and the sequence that gets methylated is C followed by G. and this is paired with itself. The other strand goes in the other direction, so it's also C paired with G, and this gives rise to the heritability of the mark. This is a methyl group, and it occurs in pairs because there are two CGs together, and usually both are symmetrically methylated. So the question I started off with, what on earth is this for? It's been known about for a very long time that there is a fifth base in the genome, 5-methylcytosine, and the question one wants to answer is, why is it there? What is it good for? So if you like, it's an actor looking for a play. The DNA methylation is added after the DNA is replicated. It's not there as part of a substrate that's incorporated into the DNA, and it does not alter the genetic code. So you still have a nice pairing between G and C and A and T, and that's not altered by this. So my involvement in the whole area sort of started with learning how to map where the methyl groups were, because it was known there was a fifth base, but no one had the faintest idea where it was. And I used these ribosomal RNA genes of a frog and used restriction enzymes to try to cut them. And so this involved a chance meeting in Zurich with Hamilton Smith. Ham Smith is famous for discovering the utility of type 2 restriction enzymes, the ones that cut at a specific sequence. He arrived in Zurich on sabbatical. First thing he did was make HPA2. He incidentally won the Nobel Prize for that a few years later. This individual, Ed Southern, was also there, famous for the Southern blot. And the three of us had tremendous chats about the new technologies that were emerging, restriction enzymes, agarose gels, which then were revolutionary. And I decided I needed to get into this, and so I ran an agarose gel of the only thing I had, which was ribosomal RNA genes from the frog. There was no cloning in these days. You just had to purify genes physically using repeated equilibrium cesium chloride gradients. And so I had some from blood, and I had some from eggs. And so I digested, this is the sort of digest. This is digesting the DNA with this enzyme, HEPA2, which was discovered by Ham Smith. He made it in bucket loads and gave me some. I cut the chromosomal DNA, and it was stuck near the top. And I cut the amplified DNA from the frog egg, and it was minced into tiny little pieces. But it's both ribosomal genes. They're supposed to behave the same way. If you use an enzyme like this, HEY3, then it does cut them both into tiny little pieces. But these two enzymes also show this dramatic difference. And of course, the reason is that the enzymes can't cut when the DNA is methylated. And the chromosomal DNA is heavily methylated, as Don Brown and Igor David had shown before, whereas the amplified ribosomal DNA from the eggs, by chance, doesn't have any methylation at all, and so it gets cut into tiny pieces. And so using this information, one could, in principle, and in fact in practice as well, map where the methyl groups were. And this is the first map we made. This is ribosomal RNA genes, the 18S, the 28S gene. There's an unmethylated region near the transcription start site, and there's another one down here. So that's the beginning of mapping, and once we knew how to map where the methyl groups were, then you could ask questions like, is there a pattern? And so this was the sort of phylogenetic phase when no marine or land animal was safe because we wanted to make DNA from it and cut it with these restriction enzymes. This is sea anemone, and in all invertebrates, apart from a few that don't have any methylation at all, you see this pattern where some of the DNA is resistant to these enzymes and the rest is cut. And so we then started looking at vertebrates, and vertebrates, oddly, always look different from all the invertebrate animals. You need to remember, despite the fact that we are vertebrates, most animals are not. More than 95% of animals are not vertebrates. So most animals look like this. They have methylated DNA and non-methylated DNA, but vertebrates, and this just happens to be the chicken, has a boringly solidly methylated genome. Or does it? We then decided to look more carefully and label the ends of the fragments. And if you label the ends of the fragments on this gel, now there's something you can't see if you just stain it. because it's a number average, number distribution you're seeing rather than a weight distribution, there's hardly any DNA down here, but it has a lot of ends because it's cut into small pieces. You see this lump here, which looks like an artifact, but actually whichever animal you use, and this is a sort of, looks like a recipe for the soup in Macbeth, but it's actually different vertebrates, they all have this blob. And this blob is derived from CPG islands. So there is a pattern in vertebrate genomes. There are non-methylated CPG islands, but the bulk of the genome is methylated. So if you look at the DNA methylation pattern in vertebrates, it's really rather boring. It's quite high all the way across. This is a neuron. This is the frequency of methylation. This is glia. This is liver. Your heart, although people emphasize differences, they're not that wildly easy to find, except in certain restricted regions of the genome. Instead, you see these non-methylated regions where the level of methylation drops down. There's a CPG island. Actually, all of these are CPG islands, and they're all, the level of methylation goes to 0, but we're looking from so far away you can't actually see that. So the CPG islands then have no methylation at all. So let's look more closely at the CPG islands. These are genes that don't have CPG islands, and each vertical line here is a CPG. So this is a representation of the sequence in which we only look at the CPGs. And you see that the CGs are rather rare here, and these are all genes that are tissue-specific. They're expressed in one type of cell only. But the vast majority of genes, well, vast majority, about 70% of genes, have a sequence like this, where suddenly you get a high increase in the CPG density, and these sites are not methylated. These are all methylated. These are not methylated over this particular region. And so the reason why I'm introducing this is because this is a key feature of the DNA methylation pattern, and it is associated, this is the start of transcription. In very great majority of cases, if not all cases, transcription starts in these regions. So if you look at CPG islands, you can see they occur at the promoters of genes. Each of the genes here, some go from left to right, others go from right to left. They all have a CPG island. If you look at the initiator form of RNA polymerase, it also sits there. And if you look at this histone mark, trimethylation of lysine 4, you also see it in exactly the same place. So the question is, what are these CPG islands for? They lack methylation. What are they good for? And the answer started to come. David Skolnick discovered a protein in quite a long time ago, about 2000, which binds to CG. And it just sat there in the literature, really, without being associated with CPG islands. And we then decided to ask whether or not it was enriched at CPG islands. And there's CFP1. It is enriched at the CPG islands, too. So why should we be interested in CFP1? The answer is because it's part of a complex that puts on this mark. And so it binds to non-methylated CG, and then it catalyzes the addition of this mark on these histone tails. So you knock it out, and all the lysine 4 methylation goes away. That doesn't really tell you too much. A more subtle experiment is this, where you insert a piece of junk into the genome. There's no biology. We've done it with computer-generated sequences that have no biological significance. And this one isn't that, but it is a bacterial sequence, but it works with everything we have tried. You insert a piece of artificial CPG island-like DNA, and you then ask, this is the CPG density, up it goes, down it goes, just like at a real CPG island. And then you ask, does this now attract CFP1? And the answer is it does. It's non-methylated. It attracts this protein. Now, what about H3K4 trimethylation, this mark that is associated with active promoters? And there it is in blue. You get H3K4 trimethylation as well. So have we accidentally made a gene? The answer is, there's RNA polymerase. No. There's no RNA polymerase there. So just the presence of non-methylated CG influences chromatin structure via this complex and actually via other complexes that also bind non-methylated CG. DNA sequence here is determining the epigenome. It's if you like, sometimes the epigenome is seen as a slightly parallel universe that has its own rules and regulations, but here it's just the DNA sequence that is telling it what to do. And other CG binding proteins are also recruiting, and several are now known from work from other labs. And this may be a key function of CPG islands, to set up appropriate genome structures, genome structure at promoters. So these are so-called CXXC proteins that recognize CG, if you like, and they bind CG, and virtually all of them create a sort of promoter-friendly, that's the sun coming out, which in Scotland is a very evocative thing. They create this promoter-friendly environment. And they come to a methylated CG and they can't bind. So there's a way in which you create a platform that is promoter-friendly. So now that's CG. What about the rest of the genome, the methyl CG, which most CGs are methylated, as I showed you in that wall-to-wall pattern. So what about them? So we actually worked on that before. We worked on the non-methylated CG binding proteins. And this shows the sort of experiment that got us into it. This is from an old paper. What we did was make proteins, extracts from nuclei of proteins, and then ask if they bound to non-methylated DNA or methylated DNA. And what we found is that there was something that bound to non-methylated DNA and methylated DNA, but if you put in a competitor, you could get rid of these things. And in here, for example, we put in a bacterial genome, Micrococcus luteus, Lysodecticus, or E. coli DNA, or Drosophila DNA. These are all non-methylated, and this complex with the methylated DNA persists. If we put in genomes that are heavily methylated, like Xenopus or mouse or cow, then they abolish this interaction. So there was something in here that binds methylated DNA, a methyl CG. And then we asked, what about transcription? And this is the most data-rich slide in my presentation, you'll be pleased to hear. If you take a reporter gene and you put it, it was the alpha globin gene, and you put it into an extract that contains these proteins, then it's expressed if it's non-methylated, but if it's methylated, it's silent. And if you then add a competitor, a bacterial DNA, it doesn't change anything. It's still not expressed if it's methylated. But if you methylate that competitor, so you're soaking up these methyl binding proteins, back comes the expression. And here's another example. If you don't methylate the competitor, nothing happens. But if you methylate the competitor, you restore. So whatever it is that is in these extracts that is able to bind methylated DNA, it is repressing transcription. And so one has this sort of picture where you have something that now binds methyl CG, and it's a different color. It's rather a darker cloud because it's repressive. And now the fog moves in, and you generate a promoter unfriendly environment. So that's the sort of high-level view of how we started off our interest in this area, these methyl binding proteins. With DNA methylation, we're used to thinking of it as a repressive. How does it repress? That's the sort of question that we wanted to answer. So the first protein we managed to purify was MECP2, a DNA methylation reader, and we found it in extracts by incubating proteins run on a gel with either methylated DNA or unmethylated DNA, and we found a protein of approximately 84 kilodaltons, or apparent molecular weight 84 kilodaltons, which binds to the methylated DNA, but couldn't bind at all to the non-methylated DNA. So was this one of those readers that was influencing gene expression? This just shows that the interaction with methylated DNA is robust. You can crystallize the DNA binding domain of this protein. There are the methyl groups in the major groove, and here is the methyl binding domain, and you can tell in atomic detail just exactly how it recognizes the methylated DNA. So what is this MECP2 protein? It's a nuclear protein bound to chromatin. It's expressed broadly. It's not just expressed in the brain. It's expressed everywhere. But it's particularly highly expressed in neurons. It's very highly expressed there. So this is when Huda's findings burst onto the scene. We were happily working on DNA methylation readers, how do they control gene expression, when suddenly the paper from Amir appeared telling us that Rett syndrome, MECP2 deficiency, MECP2 mutations are causing Rett syndrome. And we've heard a lot from Huda about this. We've also heard about the A140V mutation, which is one of the recurrent X-linked intellectual disability mutations, and we've heard about MECP2 duplication syndrome. So I'm not going to dwell on those, but suddenly, as well as having a reason to study this protein because of its as an epigenetic reader, we had a clinical motivation. Huda has told you about the importance of X chromosome inactivation. I'm just going to show you the females with Rett syndrome are heterozygous. They have a mutant copy of the gene, most of the time from the father. And then this then following random X chromosome inactivation gives you cells, a mixture of cells that are either positive for MECP2 functionally or negative for MECP2 if they have the mutation. And this is visualized here. This is the dentate gyrus of a mouse. And you can see that we've tagged the MECP2 protein with a fluorescent marker, green fluorescent protein, and you can see the interspersion of the different X chromosome inactivation patterns. So they're very tightly interspersed. It's not like the calico cat or the tortoiseshell cat where you have big patches of fur. In the brain, the patches are tiny. So we made a mouse model. We were making a mouse model to understand how the protein worked. Now we needed to make a mouse model to understand Rett syndrome as well. And as Huda has mentioned, this recapitulates the human situation pretty well, pretty convincingly. Green means they're happy, and these are wild-type mice. The male null mice survived for a while, then they got chronic neurological symptom or severe neurological symptoms which got worse and worse, and then they were all dead, usually by something like the age of 12 weeks. And they show this hind limb clasping. It's delayed onset again, but if you look hard enough, you can find things earlier. But in females, we thought there was absolutely no phenotype because you could breed these animals, and we thought, oh, this is not like the human situation. And then when you get to about 6, it varies, but 3 months, a few months of age, up to 12 months sometimes, you get a sudden decline, and you get a chronic neurological phenotype, and this lasts for the normal lifespan. So as in humans, in females, which have this mosaicism for the mutation, you get this delayed onset and then a chronic phenotype. This would be the model for Rett syndrome. So the MECP2 mouse is a convincing model for Rett syndrome, and also remember we want to understand how DNA methylation works in this way too. So it's got a phenotype, one can study it. So in passing, we were aware that the pathology of Rett syndrome involves a somewhat simplified neuronal pattern. This would be wild type, and this would be a diagram of a neuron in the mutant. It's due to, Rett syndrome is due to the lack of MECP2 in the brain. We know that because if you knock out the gene only in the brain, you also get the same sort of phenotype. And in fact, we recently knocked out MECP2 in everywhere except the brain. So there was MECP2 in the brain, but no MECP2 everywhere else. And these mice are essentially wild type. And this was published recently. Adult mice need MECP2 for survival. And this is, Huda's lab initially showed this, and we also have shown it, that if you take away MECP2, things get bad very quickly. So it's not as though this is a protein you just need for development, and then you don't need it anymore. You need it the whole time. And finally, it's just to point out that it's predominantly a neuronal disorder. There is a role for glia, but it seems that the primary cell type that is affected by depletion of MECP2 is the neuron. So the question that this begged is, since the neurons are not dead, Rett syndrome is not a neurodegenerative disorder, so if you put back the protein, what happens? There's no cell death been reported in either patients or the mouse model, or no increased cell death. The neurons are small and somewhat simplified, so are the symptoms reversible? And it's important to stress that the expectation is that it's not, because neurological disorders, in particular ones labeled as this was for many years a neurodevelopmental disorder, the assumption is that they would not be reversible. So we took the MECP2 gene and we inserted into it a stop that we could subsequently remove via Cre-Lox sites. This then stopped gene expression. The mice grew up as though they didn't have any MECP2. And then we removed that stop when the mice were ill, and then gene expression was restored. And Jackie Guy did these beautiful experiments. So now, coming back to the picture of the mouse phenotypes, we want to wait until the male mice are terminally ill. They're going to die in a week or two. Then we switch on the MECP2. And in the females, these are the real model of Rett syndrome. They're no longer young and plastic. They're quite old. What happens if we switch it on in this stage? So I'm not going to go through this in excruciating detail, but I just want to show you this is the phenotype of the mice. That's the phenotype of a mouse that has grown up thinking it doesn't have the MECP2 gene. It's just that we've stopped it, in fact. It does. And you see this tremor. You see the breathing arrhythmia. If you look at the flank, you can see it's breathing, then stopping, which is one of the features. And this mouse, it doesn't move. This mouse we would need to kill under our animal license within a week or at most 2. So that's the day it was treated with tamoxifen, and then this is the day, this is a month afterwards, and it's robustly restored to health. So it's not absolutely perfect. It wouldn't win any prizes, but it's nevertheless incredibly well and lived for over a year. And this has been reproduced with our mice in multiple laboratories. And what about the older mouse? So these two mice, this is a female. These are females, both. One's wild type, one's reversed. And this is the one that was the control. It was unable to respond to the tamoxifen for genetic reasons. And so this is what this mouse looked like before it was reversed. Obese on this background, immobile, various other phenotypes, which were all reversed upon reactivation of the gene. And these are the apneas: breathe, breathe, stop, breathe, stop, breathe, stop. And this is after reversal, that goes away. So this is quite an impressive reversal. Before I get on to the function of MECP2 again, I just want to say that we collaborated with Gail Mandel to try to see, and I emphasize the word try, to see if we could use this to convert this to a therapy, because the way we made these mice was a series of genetic tricks. And with Gail and Hélène Cheval in my lab, we obtained viruses, and this was primarily led by Gail Mandel. We used AAV9, an adeno-associated virus, put in the MECP2 promoter, driving the MECP2 gene, and then injected this into the tail vein of mice, because remarkably, this virus can cross the blood-brain barrier. Admittedly, most of it doesn't, but some of it does. And these are females who are symptomatic, so this is the mouse model of the human disorder. And although I'm not going to show you the behavioral results, the mice improved. This is they didn't actually disappear when treated. That's a pity, because that's a mouse with the phenotype. What are you betting is going to happen when I press that? Oh. Actually, as you can see, they got a lot better. So this is there is quite a significant improvement. It's not as good as an experiment as what I showed you, at least in the film, because that's not the same mouse, but, because they were done at the same time, ones that have been treated and ones that hadn't. But it's promising. It's not practical as a therapy, but it shows that at least if you have this dosage problem that Huda alerted to, you put in too much, alerted us to, you have as big a problem as if you don't have any. But nevertheless, these animals did dramatically improve, and if you can go to the website where this paper is and look at the real results in a way that is not Alice in Wonderland disappearing films. So, that is a plausible way forward with this disorder. Another way forward, so simply put back the gene, and ideally what you would like to do is edit. You would like to rewrite the piece of the genome with the mutations as you would for many human disorders. And obviously now with the advent of CRISPR, we can dream of editing, rewriting mistakes in the genome. But we are not there yet by some way. So another way of doing it is to see how this protein works, and this brings us back to the mechanisms that I was talking about to begin with. And one of the resources we have in trying to understand how the protein works, to see if we can use that information to generate therapies, is to look at the mutational spectrum. This gene is now on the beaten track for looking at in cases of developmental delay. And so children are routinely sequenced, and there are now large numbers of mutations. And these are the mutations, missense mutations, I stress missense, one amino acid has been substituted by another, that are present in Rett syndrome. And then these are mutations with, quotes, no clinical effects, at least they do not cause Rett syndrome. And you see the distributions are very different. Here we have mutations over this domain. This domain is the DNA binding domain. MBD stands for methyl CpG binding domain. It's the region I showed you the crystal structure of. And then this region here, which is also highlighted with mutations, and then a couple of mutations elsewhere. But the distributions are very different. So that then tells us that these two regions, we need to know what they do. And we do know what they do. This one binds DNA, and most of these mutations interfere with that binding. So I'm not going to go through all of the data on this. I'm just going to say that MECP2 is a sequence-specific DNA binding protein dependent on cytosine methylation. This is more or less accepted by everybody. The methyl CG binding site occurs approximately every 100 base pairs in the genome. So this is not like a transcription factor that goes to special places, drives certain genes, and ignores the others. It's spread all over the genome, and it's abundant enough to almost cover the genome in neurons, and so its distribution is sort of undulating, but very uniform, relatively uniform. In neurons, global binding is increased and redistributed somewhat by binding to a trinucleotide, methyl CAC. So this is, in neurons, really almost exclusively, there is methylation in not just CG, but methyl CA, and predominantly methyl CAC. And MECP2 also binds to that. And this, from a number of studies, is turning out to be an important addition to the binding site repertoire of MECP2. And then there's hydroxymethyl CG, and I just put this in for any of you who know about this report that MECP2 binds hydroxymethyl CG. In fact, several groups, including our own several years ago, showed that MECP2 doesn't bind hydroxymethyl CG. It does bind hydroxymethyl C in this context, but there is reportedly very little of that. So this is the binding sequence, methyl CG, methyl CG, methyl CG, methyl CG. It doesn't matter what follows the methyl CG. MECP2 binds it, that's why it's red, and methyl CAC, and a little bit methyl CAT. These are the targets for binding in the brain. So that's what the DNA binding it looks like, and many of the mutations that cause Rett syndrome interfere with that. So then what's this for? This region here, it turns out we call it the NCOR interaction domain because it binds to the co-repressor NCOR. So NCOR, for those of you who don't know, is a one of these very large machines whose structure are completely mysterious at the moment. They have histone deacetylases that remove acetyl groups from active chromatin regions and thereby repress transcription. And this one here is the one that interacts with MECP2. It's recruited by other things. These complexes don't know where to go. They have no DNA binding domain, so they need to be told. If you make a mutation that interferes, so this one here, the last of the four, R306C, about 5% of Rett syndrome, you make this in a mouse, it creates a Rett-like phenotype. And indeed, if you look in the brain and do an immunoprecipitation, you pull down MECP2. In a wild-type brain, you pull down NCOR, you pull down TBL-R1, which is a subunit of that NCOR complex. You pull down HDAC3, which is another subunit. Whereas in the R306C mouse, you do the same experiment. You don't get any of those things. You've destroyed that interaction effectively. So what does, I'd say we don't know the structure of NCOR. We know a structure of a bit of it. It would go off a long way in that direction. But little bits of it have been, the structure has been determined. Really only the colored bits, the gray bits are a bit mysterious. MECP2 actually binds to the ends of these four domains here. And these are WD40 domains, actually. They are these beautiful barrel-shaped structures. And MECP2, you can just see these dots here, sits on the end here. And this is a very specific interaction. If you take a peptide corresponding to that region, you can purify all of the NCOR in an extract. So we now have two binding sequences which correspond to the two regions that are mutated in Rett syndrome. One is the DNA binding domain, and the other one is the NCOR SMART interaction domain. So the obvious model then, this is the simple model, is that MECP2 forms a bridge between these two, and that if you destroy this end of the bridge so it can't bind methylated DNA, you get Rett syndrome. And if you destroy this end of the bridge so you can't bind the NCOR complex, you also get Rett syndrome. So I should point out there are many models for how MECP2 works. I've been telling you about this one, but there are others. And in my opinion, they are not as well backed by biochemistry, but they nevertheless are out there. So one can't be complacent. One has to actually test how this works. And there's always the frightening possibility that MECP2 does lots of things. And it's a multifunctional hub for loads of different processes. So that's an emphasis in our lab right now, is to test the bridge hypothesis. So one of the predictions is that MECP2 binds DNA and NCOR at the same time. Another prediction of our hypothesis, that's the way science goes forward. You make a hypothesis, and then you try to think of an experiment that can destroy it. And there's something exhilarating as well as frightening about destroying your own favorite hypothesis. So Rett missense mutations outside the MBD and the NID either destabilize the protein or abolish the bridge function. That's a prediction. And the MBD, this is the most aggressive prediction, that all you need are these two domains of the protein. Everything else is unnecessary. And this is a separate thing, that for overexpression syndrome, you need the MBD and the NID. And Huda's lab have already published evidence that this last one is correct. I'm just going to show you one thing. I'm going to show you some evidence that is compatible with the idea that MECP2 binds both of these two macromolecules at the same time. It's a somewhat artificial assay. If you take a TBL1, that's the lollipop thing that it was binding to, and you put it into cells, it just goes to the cytoplasm because it doesn't have a nuclear localization signal. If you put in MECP2, it goes to where most of the DNA methylation is, namely these heterochromatic blobs in the nucleus. It doesn't go there because it likes heterochromatin. It goes there because it binds to methylated DNA, and that's where a lot of it is, only because of the sequence that's there. It also goes everywhere else. And it takes TBL1 there. If you put them in both together, if you haven't put in MECP2, you don't relocalize. If you do put in MECP2, you relocalize the TBL1. And if you put in MECP2 that has a mutation in that NID domain, so it can't bind NCOR, now MECP2 goes to those dots, but it can't take the TBL anymore. So this suggests that the interaction is simultaneous. And we are engaged heavily in many of the other prediction tests that I showed on that slide. So lastly, I want to just mention, you can't talk about binding to co-repressors and DNA methylation without talking about transcription. And in a way, the reason why there were so many hypotheses out there is because what you sort of expect, simplistically as it turns out, is that when you take away a repressor, everything goes up. So you take away something that binds throughout the genome, it no longer, the genome will break free, and transcription of many genes will increase. That isn't what gets seen. It's actually rather complicated. But an analysis, and this this is partly with the excellent data set from Huda's lab, you can see that there is a change in the amount to which genes are upregulated according to the amount of MECP2 that there is bound. You also see this with all genes, but I've highlighted here genes that are involved in neurological disorders of one sort or another. And what you can see here is that the more MECP2 that is bound in vivo, the more the gene goes up when you in the knockout. And Huda also made this overexpression mouse, and you can see the opposite. The more MECP2 is bound in vivo over here, the more these genes are repressed if you overexpress MECP2. As Huda has said, these effects are small. You are not seeing gigantic changes in gene expression for many of these genes, though you do see something big for some of them. And so there is a relationship between the amount of binding and the repression level. We looked in a different system. We have some dopaminergic neurons that will differentiate in vitro very quickly, in a few days. iPS cells take weeks. These take 5 days. You can manipulate them. We have knocked out MECP2, it's gone, or we have normal levels of MECP2, or we have overexpressed using a viral vector to very high levels, tenfold overexpression. So varying the level of MECP2, we can then look at the effect on gene expression. We can look at neuronal markers. These cells make neurons just fine, even if they've got 10 times too much MECP2. But you now look at gene expression, and this is all genes. The more binding sites you have, the more genes are upregulated when you take away MECP2. And the same, the opposite, when you overexpress MECP2. Now, the ones with the most binding sites go down the most. So there is a relationship between gene expression and the level of binding. So that's where we are with the understanding of how this protein works. It's a difficult protein to study because the effects are rather small. But I'm now going to just end by summarizing the relationship between this protein and transcription. And the first is that deficiency or overexpression in the mouse brain stimulates or inhibits transcription of a subset of genes according to the levels of bound MECP2. MECP2 occupancy affects gene domains, not promoters. I didn't show you that, but it extends quite a long way up downstream of the promoters of the genes. The changes are often small scale and gene dependent. MECP2 behaves as a global DNA methylation dependent down modulator of transcription for many genes. All transcription is reduced in the absence of MECP2. I haven't shown that, but globally, transcription goes down. This could be a secondary effect. The cells are smaller. But, and I echo Dr. Zogby's conclusions here by saying slight deregulation of thousands of genes may underlie MECP2-related autism spectrum disorders. So it's a complicated story, and it requires, I would say it's also an unfinished story. There are points that we have yet to elucidate. And so finally then, these are the people who are in my lab now. The people who've done the work I've talked about are too many to show on one slide. But this is Jackie, who did the reversal experiments, and this is Matt, who found the NID, and this is Justina and Ruth, who did the experiments with the human neurons, and I'm not showing people who did much of the other stuff. And this is Rett Syndrome Research Trust, who fund our work, and also the Wellcome Trust in the UK, who fund our research. So thank you very much for your attention.
Thank you very much for the laureates, Professor Bird and Professor Sockby, for telling us the most original research findings and also the most upfront research directions that they are taking on. And now I think we can take several questions from the floor. If you have any questions, then do feel free to ask. And the laureates, I'm sure, will be very happy to discuss with you of whatever they have in mind.
Just wonder, what do you think of the hypothesis that the increase in autism may be due to oversupplementation of folic acid affecting women who have kind of deficiency in MTHFR, affecting the methylation status? Do you think that could be the reason why there's more autism recently in a couple of decades?
I will take a stab at this question. So the question is, what accounts for the increase in autism? Could it be folate supplementation in women? I don't think the evidence is sufficiently strong in this area. Although one has to be careful, as you know, folate supplementation is to prevent neural tube defects. And just as there are vulnerable people who need the folate, there may be people who don't need as much folate. But I don't believe the evidence is strong enough to really explain idiopathic autism by folate supplementation in healthy pregnancies.
Thank you.
So I would just go slightly further and say, in my own personal opinion, there is a very strong pull in the direction of environmental influences being responsible for disorders of this kind. And one has to be very circumspect about quite a lot of the evidence. In some cases, there is something going on, but in many cases, there is appears to be overinterpretation of rather weak data. I'm not saying that's true in this particular case, but it's something generically in this area you have to be aware of.
Okay, gentleman upstairs.
Yeah, thank you for the impressive talk. I have a question about the time course. So autism patients usually have a very early onset. So why in the Rett syndrome case, it takes some time to let the patient or the mice to manifest the defects, especially for some defects like the wheelchair bound, it takes very, very long time. So how to explain that?
So it's an interesting question. Why is the progression of this disease slow? I do think it does take time for neurons to really undergo all the molecular as well as synaptic changes, either due to the loss or the gain of the protein. So it is not an on and off phenomena. It does take time. Even in the mice, if we were to delete the gene in adults, it takes some time for the symptoms to occur. And in the duplication, when we rescue, even if we normalize the protein level today, you're not going to see recovery tomorrow. It takes a few weeks. I do believe this is due to the neuron's response to the absence or presence of the protein. Now, the second question is, why do some symptoms take much longer? Why does it take years to see the motor deterioration? And I think that's also an interesting question, and I think it probably has to do with the robustness of the circuit. And circuits that are more vulnerable, it doesn't take much to see the symptoms, whereas those maybe that are more robust or have to take a lot of hit to manifest, they will take longer time to show. That's the best explanation I have from the clinical data.
So a following question maybe is, the wheelchair-bound phenomena is not due to the MACB2 affect the genes in the skeletal muscle system, while it's the defects in the neuron-muscle connection point, the defects from the brain. Okay, is that true? So the wheelchair-bound phenomena,
Yeah, it's
Yeah.
due to the neuronal function. I think in a very nice paper from Adrian, he really definitively showed at least there is no muscle phenotype. It's really, yeah.
Yeah, thank
So
you.
we generated a mouse which had MECP2 in the brain, but it didn't have any in its other tissues, including muscle. So in fact, these mice were more or less wild type. The suggestion is therefore that many of these, if not all of these, peripheral functions are controlled from the central nervous system.
Effect of loss of MECP2, which is obviously an important enzyme for basic global methylation of DNA. And I wonder whether this has anything to do whether a cell is dividing or not dividing.
Well, neurons are post-mitotic.
Yes.
So, you know, in the restoration experiments, they grew up with no MECP2, and then you put it back, and things improve dramatically. So, I don't think there's any relationship to cell division. In fact, MECP2 generally goes up when cells stop dividing. It's almost as though it's a feature of non-dividing differentiated cells, not just neurons, but other cell types as well.
So that was what I meant, was whether it was essential for a non-dividing cell as opposed to a cell that's dividing. And so, therefore, you would expect then, as particularly since neurons don't divide very much at all, don't divide. And so loss of, I'm just wondering whether loss of MECP2 is more harmful to the neuron because it doesn't divide.
Well, I think, you know, there are other cell types that don't divide very much too, but admittedly neurons are the outstanding non-dividing cell types. I think a key is that there is far more MECP2 in these cells than there is in anything else. It seems to be more important for day-to-day life of a neuron than it is for these other cell types. So I personally don't think, so while MECP2, high levels of MECP2 is a feature of differentiated cell types, I don't think its function is necessarily related to whether or not it's dividing. I think it's more important in neurons because neurons do something slightly different with it. They have an extra set of binding sites, these CAC binding sites, and the abundance is really spectacularly high in those cells.
I have a question to Professor Sohi. So in your presentation, you show some interesting data on synchrony, right? So it's an X-linked intellectual disability disorders, right, MECP2. So I just wonder whether you also see similar mechanism in other X-linked intellectual disability diseases, such as the female-limited epileptic encephalopathy due to PCDH19, which is a protocoherent gene. That means even the male get the mutation, they are normal, but the female who got the mutation, they will have epilepsy because of the circuitry problems.
So that's a very interesting question. To my knowledge, no one has looked yet at the synchrony pattern in that disorder, but it is really interesting that a mosaic brain is more vulnerable than a totally null brain in that particular disorder, and it could be due to abnormal synchrony, but no one has looked to my knowledge.
Thank you so much for your excellent talk. I have a question about the timing for treatment. So you mentioned that time window for treatment is important because we know that autism spectrum disorder is a neurodevelopmental disorder, right? So is that the earlier the better? And the second question is, because I work on Fragile X syndromes, so we also found that hippocampal dysfunction underlying the anxiety, behavior, and also cognitive impairment. So from your data showing that there's an impairment in neurogenesis and also LTP in the CA1 regions. So what I found in the Fragile X syndrome mice is there's an impairment in the mGlu5 LTD, but there's an impairment in AMD receptors hyperfunction. So it looks like that there's a regional specific. So wondering is how we can tackle this problem for the drug treatments. Because in the case of Fragile X syndromes, now clinical trial using the mGlu5 that work in animal, but actually not working in clinical.
Yeah. So this really is a very important question, and I think I've given a lot of thought to this, and that's why I mentioned that timing is important and the need of adjunct therapies such as cognitive and physical therapy. I think you're right. The mGlu-R story was really strong in the Fragile X, and the treatment worked beautifully in every organism tested, fruit flies and mice, and both genetically and pharmacologically, yet when we went to a clinical trial, there was no benefit. And one of the worries I have is that we may be missing a potentially good treatment for Fragile X because of how the clinical trials were designed. I think they were designed to give the mGlu-R antagonist, but to do it and look for phenotypes rather than say, these are young adults who have not gone through the typical way a child would learn. A healthy child goes through a whole sequence of experiences and learning so that they can speak, so they can socialize and do different things. When someone hasn't gone through all of that, even if you corrected the problem with them, it may be really hard to get an effect. You may have to combine the therapy with a lot of training, a lot of physical and cognitive therapy coupled with the targeted as mGlu-R antagonist. So I think that's something we really have to recognize. Mice are different. You might rescue things in mice much easier than in a human who has to go through that long process. And because of that, I think as we contemplate trials, the earlier we do them, the better. Because I think if you can do something in a 3-year-old red girl, it may be much easier to at least discern if there's going to be a benefit, so that if you want to go to the older girl, then you know there is hope because you've seen the benefit before the disease has set in a very serious way. Then you can do it with intensive cognitive and physical and rehabilitative therapies.
So I've got a question for Professor Bird. So you showed us some data just now on how the NCORE transcriptional is a complex interacts with the MECP2, right? And you also showed us how if you inhibit the function of this NCORE complex, you get a phenotype quite similar to Rett syndrome, right? So I was just wondering, maybe MECP2 and its function is quite specific to neurons in the brain, but what about NCORE itself? Surely that's got a wider function with a lot of effects in other cells. I mean, when you inhibited that function, did you see any other systemic manifestations, and how does that relate to maybe other diseases we know?
So, I mean, it's an interesting question. In fact, we mutated the MECP2, so it couldn't interact with NCORE, but everything else that NCORE does was still okay. But that doesn't mean it's not possible that you've got a very abundant protein that normally interacts with NCORE. It's possible that when it's no longer there, the balance of things that NCORE does is disturbed. Now, I think that's a very interesting possibility. It's not an exciting possibility, but many of these, but quite often things that are true are not necessarily the most exciting things. So, I mean, there may be a germ of something important there about the imbalance between what MECP2 does with NCORE and what everybody else does, all the other genes do with NCORE. There may be something to follow up there.
Because of interest of time, this will be the last question.
No, thank you. Well, my first comment before my question is thank you both for wonderful lectures, both complimentary. My question is probably for Adrian Bird because of he did touch on the multifunctional role of MECP2, and you had really pioneered not only MECP2 but the MBD family members. The role for MECP2 is methylated DNA is well described. What are your thoughts on the role of MECP2 binding to other nucleic acids like RNA, for example, and potentially regulating genes with chromatin?
Yeah, so, I mean, we have been led, I would say, by the mutational spectrum, which points to those two particular regions. Now, you could say those two particular regions are important, but not necessarily for the reasons that we followed up. So yes, it binds DNA, but maybe it binds RNA as well. Well, yes, it binds NCOR, but maybe it binds other proteins too. So that's something you have to be aware of. And MECP2 does bind RNA like gangbusters. It's many years ago, decades ago, we put MECP2 on a column, we poured through an extract, all the RNPs in the cell came out. But it turns out if we treated with ribonuclease, they all went away. So it was binding RNA. But we have not found it very easy, and another lab has published as well, to find which bits of the protein are required for RNA binding. And the thing about RNA is it's a sort of big string of acidic residues. And the thing about MECP2 is it's a basic protein, and bases and acids stick to each other. So you have to also try to distinguish whether or not this lovely looking interaction is just an accidental artifact. So that's why we rely on the genetics to tell us where are the mutations, what interactions there are. And we've not found, we've not actually tested it very carefully, but the RNA binding doesn't seem to be interfered with by the mutations that cause Rett syndrome. So therefore, while it may be important at a lower level, it's not a cause of Rett syndrome. And I have a feeling this is getting at what you're saying. There are many conserved regions of MECP2. It's not clear they all are required to avoid Rett syndrome, but they may be required for other things at a somewhat lower level of stringency. So we've tried to focus only on Rett syndrome and put everything else out of our minds.
Ladies and gentlemen, let's give a big applause of thanks to our laureates, Professor Adrian Bird and Professor Huda Sophie, for being with us today.